Good afternoon, everyone, and welcome to today's EAStalk webinar. My name is Anna Viskovic, and I'm Project and Program Manager at the European Aquaculture Society, and I will be moderating today's session. Thank you all for joining us. We are delighted to have participants from across the aquaculture community today with us. Before we begin, I would like to share a few housekeeping points. This webinar is being recorded and will be made available after the event. We encourage you to submit your questions throughout the webinar using the Q&A function. We will address as many questions as possible during the discussion session at the end. We will allocate 15 minutes for discussion. If you experience any technical difficulties, please use the chat function and we will do our best to assist you. Now let's move on to today's topic. Today's webinar explores the scientific background, production methods, and practical applications of immune-stimulating products derived from the yeast and fungal biomass. Immune modulation is an increasingly important area in aquaculture nutrition, particularly as the industry seeks sustainable solutions to improve animal health, performance, and disease resilience. Our speakers will discuss the history and functionality of beta-glucan-rich products, compare yeast-derived and fungal-derived sources, and present the latest research on Citribel newly developed fungal mycelium ingredients. The webinar will cover production quality, unique products characteristics, and application studies conducted in shrimp and salmon. Today's webinar is presented by Citribel, a Belgian manufacturer with decades of expertise in transforming sugar molasses into citric acid, citrate salts, and mycelium ingredients through natural fermentation processes. It's my pleasure to introduce our speakers. First, we have David Dehaus, Key Account Manager at Citribel. David brings more than 20 years of experience in the pharmaceutical industry with expertise spanning cardiovascular diseases, diabetes, and oncology. With a background in biomedical sciences and research at University of Antwerp, he now focuses on next-generation beta-glucans and their application in aquaculture and pet food. Following him, we have Dr. Edith Wildiers, Project Manager Innovation at Citribel. Edith has a strong background in quality management and research and development and is responsible for new product development and introduction within the company. She has an extensive experience coordinating innovation projects from concept through implementation while working closely with production, quality control, and commercial teams. Together, they will provide both scientific and industry perspective on this emerging field. Edith and David, thank you for joining us today. The floor is yours.
Good afternoon, everybody. Welcome also from my side to this webinar. And I thank you for the introduction and for giving us the opportunity to present our company, our product, and the possibility to highlight our new innovation. Together with my colleague Edith, I will first introduce our company, the product, and our production process. And afterwards, Edith will then guide you to the application in aquaculture of this new beta-glucan, one from a fungal origin. Now, why is Citribel coming with an alternative type of beta-glucan? The reason is there are and there will always be challenges in aquaculture. And on the one hand, we have the growing global population, which goes hand in hand with the growing demand for the human consumption of fish. In 2025, so last year, 75% of the human consumption of fish came from aquaculture. Since the early 1990s, there have also been restriction in use of antibiotics throughout certain regions in Europe and the world. And also there's some water pollution, and as you might even experience yourself right now, temperatures are rising not only for us, but also for the seawater. And that leads to beneficial or favorable condition for some pathogens. On their turn, these pathogens, they lead to economic losses and revenue losses for the farmers. For example, in shrimp farming, every year globally due to white spot virus syndrome, around $1 billion is lost. And for salmon farming, the Tenacibaculum and the Moritella, both ulcer diseases, for every farm each year, it's about 1 to 2 million loss in dollars. And for Moritella, globally per year, hundreds of $1 million in revenue loss. Now, who is Citribel? Citribel is a circular company, so we use as a starting point the waste stream of the sugar industry. Of that sugar molasses, we applicate a surface fermentation. We're going to see later on how that actually goes into place. And after the surface fermentation, when the sugar molasses is extracted, is converted to citric acid, and we have our mycelium, then we get biotech products, citric acid, citrates, mycelium, the low-K vinasse, gypsum, and all other kinds of salts. At the moment, or at present, Citribel is globally known as a citric acid and citrate producer, but we also have, due to the manufacturing process of that citric acid, we have lots of amounts of mycelium, and the mycelium was primarily used up till now as an animal feed application. However, by the main challenges in aquaculture and the strong results observed in animal nutrition, we aim to shift to the future to more get the strength of our mycelium and have this as a main product to work with our biotech products at the end. How is this surface fermentation actually, how does it work? You see here on the right side of the screen, you see all the trays or dishes, as you would like to call them, that's up to you. To maximize, as the name says itself, surface fermentation, it's key that we maximize the surface to grow our mycelium on. So each tray is individually filled with the sugar molasses and is then incubated with our Aspergillus niger strain. And at the course of about 6 to 7 days, you see the Aspergillus niger producing, with the fermentation of the sugar molasses, the large mycelial mats that have a unique biomass composition. It's a non-GMO strain with a high beta-glucan content. As we go more into detail in the compounds or the layers of the cell wall of the most known beta-glucan sources, that are the yeast or the fungal sources, we see the same cellular membrane compounds, but from the first glance, we see a great difference in the manoprotein layer. Yeast cells are all the way around covered in manoprotein, as in fungal mycelium. There you can see there are far fewer manoproteins, and that's actually a key fact to have your beta-glucan bioactive or functional, because the manoproteins, they can inhibit the beta-glucans to interact or to bond with the receptors of the immune system, namely the dectin-1 and the CR3 complement receptor. Now, the difference in cell wall composition is actually the fact that we have a very different production process for the production of our new Mycofence. With the yeast cell wall, because of all the manoproteins at the outside layer, you have to have a cell wall disruption, which means you have even eventually do the physical disruption of the cell wall, do it enzymatically, or do it with alkali or acid solvents. When you use those products, you have to remove, of course, all the residuals. Then you have to purify your product and you have to dry it to get it onto the market. Now, the main advantage of our mycelium is that there is no disruption of that cell wall necessary because of the absence of the monoprotein layer. So the processing of the product is more sustainable and is more environmentally friendly. We just have to wash it with water and then purify it and dry it. So to sum up, the unique product specifics are that our Mycofence is a different structure beta-glucan with a content of more than 60%. It has a different structure and it has low monoprotein content. That's why we also have a different production process, which is more sustainable. And our product, the Mycofence, is only produced in Europe. Now the question is, we all know all the yeast-derived beta-glucans from the pharmacy and the application in aquaculture. The Mycofence, the new mycelium-derived beta-glucan, has it also the same? Is it better? Or as it has a different functionality? And this is part 2 that I'm going to give the word to my colleague.
Thank you very much for the introduction, David. In this part of the presentation, I will further focus on the functionality of the Mycofence as compared to the yeast benchmarks. So we have done pathogen challenges on the one hand with shrimp and on the other hand with salmon. We have chosen for shrimp because they do not have a specific immune system. So actually they are more sensitive to infections and beta-glucans are standardly used in their feed. For salmon, of course, everyone knows that production cost is very high, and there also the pathogens should be eliminated. So there also the immune stimulating products such as beta-glucans are standardly used. Now, our first trial has been done in cooperation with Imaqua here in Belgium, and it's a very well-known research institute. In total, 5 different groups were compared with each other. We had Mycofence at 2 different incorporation levels, 0.1 and 0.3, and then also 2 different benchmark groups. Of course, the purpose of this trial was to evaluate the efficacy of the Mycofence as compared to the benchmark products, on the one hand on the growth, and on the other hand on the resistance to the white spot syndrome virus. So as you can see in the trial design, the trial lasted in total 38 days, of which 28 days of feeding and then 10 days of challenge. And the parameters that are recorded as an output are growth, feed intake at day 0, 14, and 28, and then mortality at the end of the challenge. And yeah, this is a very important result already. There was no difference in weight between the different groups, which means that actually the Mycofence is also safe to use, because often it's stated in literature that a lot of energy can go towards immune stimulation when using such products. However, this was not the case, whereas we did not see a negative effect on the feed conversion rate. As a contrary, there was a clear significant decrease in mortality. So the model was designed to an end mortality of 50%. And this was statistically significant decrease by adding Mycofence to the feed at an inclusion of 0.1, but also at an inclusion of 0.3. And this decrease was also very large. It was even with 48%. Now, still remains actually after this trial a question towards the optimal incorporation rate. So therefore, we have done an additional trial at Kasetsart University in Thailand, where additional incorporation rates were included. So Mycofence at 0.1, 0.15, 0.2%, and again, 2 benchmark products. So here, the purpose of the trial is very similar, of course. It's again to evaluate the efficacy on growth, but also on resistance, not towards a virus, but towards a bacteria. And this is Vibrio. And the test setup is very similar. So there are 30 days of feeding in this trial and then 10 days of challenge. And as an output, growth, feed intake, and pathogen load were recorded after the feeding, and then mortality at the end of the challenge. Here, the model was designed towards an end mortality of 60%, and that was reached for the control, which already shows that the trial was performed in a correct way. And then by adding Mycofence to the feed, we could see a clear reduction. Already for an inclusion of 0.15%, mortality was decreased to 40%. So both inclusion of 0.1, 0.3% of Mycofence, but even 0.2% here also showed that there was a clear resistance towards pathogens. And this was not found by the benchmark product. In addition, actually the same results were found for the pathogen loads, a significant decrease in Vibrio from 40 to 10 and even 20 with the addition of Mycofence. So as a general conclusion, we can clearly state that inclusion up to 0.2% of Mycofence did not impact the growth, and that the incorporation of 0.15 and 0.2 resulted in statistical reduction in end mortality for two pathogens, which is the white spot syndrome virus and Vibrio. Those results were also found for the bacterial loads. And we actually also have blood parameters, which I will not discuss here in detail, but those results can also be shared afterwards. Now, the second part of this presentation will discuss the outcome from the in vivo trials with salmon. So therefore, we have cooperated with ONNA, a well-known research institute in Canada, where the Mycofence was also included in similar incorporation rates, and again compared with benchmark yeast. In this case, we have done a dual challenge. So two pathogens could be included in one trial. The feeding phase lasts, of course, longer for salmon. It's 5 weeks, and then after which also a challenge is done for 7 weeks. And the two pathogens are Moritella, viscosa, and Tenacibaculum, so both causing winter ulcer diseases. And as an output, growth parameters were recorded at the end of the feeding and mortality at the end of the challenge phase. Now, also for salmon, very important to notice, no impact on growth was seen. The weight and the feed intake were very similar for all different groups. However, for end mortality, we could see a clear impact. The end mortality of 70% was reached for the control group, and this was decreased statistically for an inclusion of 0.3% of Mycofence, and also no statistical difference was here seen for the yeast benchmark. Now, for the Tenacibaculum, we already saw for 0.2% of incorporation that there is a statistical significant decrease. So this is even lower concentration than for the Moritella. If wanted, all the details from this trial can also be read in the paper, whereas we have a publication on this trial. This is publicly available and can also be looked up with the link on the slides. No, actually, after all those pathogen trials, the only question then that remains is if this product is safe to use for a long period of time. Whereas certainly in salmon production, the product is added in continuous feeding, often for long periods. So there it's not wanted. It would have a negative impact, of course, on growth in a long time frame. So we have done a feeding trial at ONNA to check upon the growth during 91 days. And the parameters measured here were bulk weights at 0 days, 7, 28, 63, and 91 days, and then blood samples taken at 91 days. Here you can also clearly see that the product is safe to use. So the differences, there are no differences in weight between the different groups. And yeah, the general variation for salmon at those different ages in the feeding periods is 15 to 20%. And this means that actually the differences fall within the variation as seen here. Now, also blood parameters were followed up, but we did not see any differences between the different groups. We will not discuss those in detail here, but they actually confirm that the fish are still in good physiological conditions and that the product is thus safe to use for longer periods of time. So we can also share like the details if anyone would be interested in them. Here, as a general conclusion from the salmon trial, we can state that inclusion of Mycofence resulted in a statistical significant reduction in mortality for 2 pathogens, Moritella and Tenacibaculum, and up to 0.3% of Mycofence did not impact growth parameters at all, and also not blood parameters. So just as a summary here to conclude, yeah, we are very happy today that we have been able to present to you all details from the go-to-market trajects, actually. Mycofence is an immune-stimulating product. It has been proven by several trials. It has a beta-glucan content higher than 60% and a mannose concentration lower than 1.5. And we are able also to produce it with a circular natural fermentation process, which actually proves the sustainability as well. And in addition, it's GMP plus guaranteed, so quality certified. Yeah, I hope you all enjoyed our presentation, and thank you very much for your attention.
Thank you very much, Edith and David, for this insightful presentation. We will now move into the Q&A. So I would like to remind you all, if you have not yet submitted and we didn't receive any questions so far, please do so using the Q&A box. We will try to answer as many questions as the time allows. So to get us started, I have prepared some of the questions. So what do you foresee for the coming years towards production volumes?
For now, the production volume of our Mycofence is 1 ton a year, but now we are still using our pilot facility. When in the future we're gonna expand to the volume that we want, it goes up to 5 or 6 tons a day of mycelium at an incorporation level of recommended 0.2%. That means you can actually make about 3 tons of 300 tons of food every day for the aquaculture world.
Yeah, that's quite impressive. I would like to remind as well the audience that you can note the Edith's and David's email addresses. So they have it shared here with us on the last slide. So make sure that you know it somewhere. And if you have any further comments or questions for them, they will be really happy to give you an answer on them. The second question I have for you is, why does this project fit within the sustainable aspect of the company Citribel?
To show you that, I'm gonna go through the beginning of the presentation, and I'm gonna use this slide. The sustainability is actually based on the product that we start our fermentation process with. So here we have the sugar molasses. It's a waste stream from the sugar refinery. And actually, as a quality standing point of view, it's also interesting to know that we don't use one particular source of sugar molasses, but we always start our surface fermentation process with a blend from different supplier of the sugar molasses. So if we always start with the same blend of products with the same characteristics, we can all assure that our end product meets the quality that we want it to have. So we only use the sugar molasses as a waste stream that we validate through the surface fermentation. Main product was citric acid for Citribel, but now with the mycelium that we had good results with in the animal feed and the challenges in aquaculture, we can give also more of the mycelium back to the environment through the feed applications of aquaculture.
Thank you, David. We just received a question from Devika Suresh. Is there any ongoing studies on other species as well, tilapia and others? And what about the results in early life stages?
Thank you very much for the question, a very interesting one. What is important to notice, and I think we have not yet mentioned it in the slides, is that we are able to produce the product with different particle sizes, and they also have an impact on digestibility. So for early life stages, it's often requested by feed producers to have products that actually have very small particles. That is already possible. So yes, the Mycofence could indeed be used for those species. No, we have not yet tested the product for very early life stages. However, there is a lot of potential there. And yeah, we hope actually indeed to do such a trial in the future, because there, of course, it makes a lot of sense to have to have the immune system stimulated, for example, in hatchery feed, whereas the cost is very high, and also a lot of such products are already used. For tilapia, we indeed know that there is certainly also a request for such products. However, for Mediterranean fish or fish that are produced in Mediterranean areas, we have not really been focusing on, but we indeed in the past have looked into the potential. But actually there it was more into the protein content. So actually the Mycofence is produced from a biomass. The biomass has, of course, the certain protein content as well. For the tilapia, so we have done indeed a tilapia trial where the biomass was compared with the control group, and where it was shown that there is also potential for the protein that is contained in the biomass, that the tilapia grows actually more than just the control group. This was done in an open pond trial, which means that there is a lot of variation. So these results actually are really indicative, yes. I hope that answers the question. If there is more information needed, we can also share it afterwards.
You're really well prepared. Thank you for this. The next question we got is from Mariana De Stefani. Hi, Ariana, nice to see you here. Hi, everyone. I would like to ask if any mucus analysis has been conducted, and if they saw an interaction between pathogens and mycelium at cellular level. Do you plan to test further agents, for example, amoebis, viruses? Thank you, and amazing work. Thanks, Ariana.
Thank you very much for the question. Also a very good question, whereas we have been thinking about the impact on the mucus layer when doing our last ONNA trial, so the feeding trial where the Mycofence was fed for 91 days. And it's stated often in literature that there is an impact on the thickness of the mucus by beta-glucans. So it would be really very, very interesting to have that one tested. in addition, we think that by indeed changing this mucus layer, there are functions related to that, like, for example, injuries caused by fishing nets or so on. It's also often mentioned in literature that injuries could heal better. And that also when there are a lot of sea lice, which is a very common problem, of course, that they can attach more difficult to this mucus layer. So actually for both problems like injuries and sea lice, this would also have a lot of potential, of course. But yeah, we have not yet the data today. So indeed, I think it would be a very good recommendation.
Thank you, Edith. Thank you, Ariana. The next question I have for you is, where is the product already registered?
The product is, for the moment, it's registered in the EU, and in the United States, we have an AFCO definition of our mycelium, and it's the extracted press cake category, 36.2. In Chile, it's registered as a feed and aquaculture product. And Thailand and Ecuador, that's now I'm working on contacts with local distributors, because for those two countries, we have all the paperwork in order and all the testing done, but you need to have a physical address to get your product registered over there. So I'm just waiting for a green light of those two distributors, and then it's available in Thailand and Ecuador as well.
Perfect. Thank you so much, David. The question we got now from the audience is from Kalemi Violetta. Is there a publication available for the trial on tilapia? The one that you just showed.
No, I'm sorry, that's not now. But if there is interest, we can certainly set up a meeting to go through the details of the trial. Yeah, that's certainly possible, but there is no publication.
Perfect. Thank you so much for the opportunity for the attendees to have a meeting with you. So please, Violetta, make sure that you note the Edith's and David's email addresses so you can get in touch with them later on. The next question I have for you is, what are the physical characteristics of the product?
When the product is commercialized, it will be available in two types of packaging, one big bag packaging of 250 kgs and smaller bags of 15. And inside those bags, you're going to find a product with a dry matter content of above 93%. We have then the particle size recommended of 100 micron. The density of a cubic meter is about 5 to 700 kilos. And the shelf life for the moment is 24 months that is labeled. But the consistency and the stability of the product is still ongoing. I think, as a matter of fact, we are now at about 26 to 27 months. In the meantime,
yes.
The beta-glucan content is above 60%, with a very low monoprotein or mono-oligosaccharides of less than 1.5. It appears as a light brown free-floating powder, which is odorless.
Perfect. And it's actually quite great that you had in mind also the smaller scale buyers. So you have the bags prepared in also a smaller quantities. What we received now is the question from Devika Suresh: how much aligned is the product with EU organic regulations to be used in organic feed production?
Yes, also a very good question towards marketing, very important one. we have checked upon the natural status, which is possible to have this claim mentioned for some countries. However, the organic label, we also have to still search for the details that are underneath this regulation. And yeah, I think that's certainly, as David mentioned in the introduction, so for the production process, indeed, we have a mild processing step in which the washing is done. But yeah, there is a low concentration of chemicals used there. And yeah, I think it will matter if this concentration is below the concentration that's mentioned in the regulation or not. And I do not have the answer yet today for that, but I just wanted to mention here that we indeed hope that that would be possible.
I could ask the people who have this question if they are thinking about the Mycofence, which is a more purified form of the mycelium, because there is also another extraction of our fungal mycelium, which is not intended to be used in aquaculture, but is more intended for use in pet food, and that's the beta-glucan, which has a lower beta-glucan content, but it doesn't have that washing step with a little bit of solvent inside. And there we can say it is natural process because there it's actually just washing with water. So it depends on the purity that you want. Do you want a microglucan with 35% of beta-glucan content? Then that's only washing with water. Do you want the Mycofence with a 60%? Then there is a little solvent step mentioned, but the concentrations are really low. So it could be country dependent on if it falls in the category yes or no.
Thank you, David. This is just a reminder that soon we will close the Q&A. So please, if you have any questions, let us know. The next question I have for you is, do you know if the product remains functional during processing? For example, if it can withstand high temperatures?
Thank you for that question. So maybe also because we were still discussing the processing, it's good to mention that during drying, the product temperature does not go above 70 degrees. So the product temperature is maximal 70 degrees. But we know, whereas afterwards, of course, there's also been an extrusion protocol to make the pellets for the aqua feeds as well, for shrimp and salmon. And in this protocol, the maximum temperature would be 90 degrees. So still, we have seen that for both trials, the shrimp and the salmon trial, that in both cases, the product was still functional. So we know indeed that the processing does not have an impact here on functionality. And also, in addition, in literature, it's mentioned that for beta-glucans, there will only be a denaturation at temperatures above 200 degrees. So that means that actually, if temperatures would stay below that temperature, that the functionality is still guaranteed.
Thank you, Edith. And what quality parameters are guaranteed upon commercialization?
So for the quality, we of course follow up product quality. That's a very important one. Also, the first one that we have implemented. So we have implemented here in the laboratory a method to measure beta-glucan content, which is not easy to do because there are a lot of different papers on beta-glucan content and so on. But we use a method with an acid hydrolysis. It's also called salmon hydrolysis. We have done the analysis in-house and in parallel externally to check upon the validity of the methods, and we have been able to indeed validate that method. So with this acid hydrolysis, the beta-glucan chain will be hydrolyzed until glucose, and this glucose can then be recalculated into beta-glucan when using a standard. And you can see here that indeed for every production batch that's produced, we guarantee this quality parameter, and that's also above 60% for Mycofence. Besides that, there is of course also process quality that needs to be followed up, and for the process, we have already shown a couple of times. We have a step where the residuals are washed out and then the drying step. So in this first step, of course, also the beta-glucan content is followed up. And then the second drying step, it's the physical characteristics of the product. So it's dry matter content, particle size, that's all followed up. And we, of course, guaranteed all those analysis with a certificate of analysis.
And just a side note, the product is also GMP plus FSA certified.
Perfect. Thank you so much. The next question I have for you is, was there an effect in the blood parameters taken during the challenge trials?
Yes, we did not have the time during the presentation to go into detail. So, and this is not published yet, but we are preparing a publication also on the shrimp trial. And you can clearly see that indeed for the blood parameters that were taken after the feeding, so after 30 days of feeding, that for Mycofence at 0.2% of incorporation, total hemocyte concentration is very high. It's actually the control times 10. So this is a very good result. This also means, because those hemocytes, actually you can compare them just with lymphocytes for a human. And that means that actually the shrimp can eliminate pathogens in a specific way with phagocytosis. And that's also seen here in the second column. So total hemocyte concentration is higher, phagocytosis is higher. And in addition, there was also an expression of two enzymes, polyphenol oxidase and superoxide dismutase, for which the superoxide dismutase actually also says something about stress resistance. So with these enzymes, oxygen radicals can be captured. So that means that for shrimp that are subjected to stress, that they are more resistant as well by this enzyme. So not only they have more immune resistance, but also more stress resistance.
Thank you, Edith. And which additional tests would you recommend to do with these products beside the challenge trials?
So indeed, I already had the opportunity to, with the other questions, to mention that the trial on the mucus layer would be a very interesting one. So I would certainly like to go for that one. But in addition, we also think that for the stress reduction, that there is also a possibility. In literature, it's often stated that beta-glucans have an effect on stress reduction. So there will be faster recovery after a stressful event that can be followed up by a long period of stress, for example, temperature stress, crowding stress. So that's a possibility. And another one that would be interesting is to have it tested as an adjuvant for vaccinations. because there can be a stronger antibody response when beta-glucans are added together with an vaccine to fish, such that the immune activation would be more.
Thank you. David, do you have anything else to add?
No, I just added the word pronounced as the vaccine response. That's the only thing.
Thank you. So as the last question for today's discussion, can you maybe please go to the last slide? So in case the attendees didn't yet write your email, they can do it now. So the last question that I have for both of you is, what cell characteristics accounts for the differences in production processes?
Then I'm going to go through the slides that you also passing by several times this presentation. The main thing that we saw during the production process is the thickness of the wall in the yeast cell with the extra layer of the monoproteins. But when we see, actually, I didn't know that. It was new for me at the beginning of my period here at Citribel. But a yeast cell is actually also a fungal cell. The only typical difference between yeast and a fungus is that a yeast cell is a unicellular cell, while a fungus is actually the kingdom of the species. And normally for fungal cells, they grow in hypha that are elongated tubes, and the cell composition isn't all a unicellular single cell one, it's a multicellular thread, in fact. And that's why the frailness of the cell membrane of a fungi allows us to use a more softly production process with washing and drying, and we don't have the harsh solvent applications that are necessary.
Thank you so much, David. So as we come to the end of today's webinar, I would like to thank you both, Dr. Edith Wilders and David Host, for sharing your expertise and insights with us. Today's session highlighted the importance of innovative feed ingredients in supporting aquaculture health and sustainability, and provided valuable scientific evidence on the potential of fungal-derived beta-glucans in shrimp and salmon production. I would also like to thank Citribel, EAS corporate member, for supporting today's webinar, and all of you for taking the time to attend and participate. A recording of this webinar will be made available after the event, and you will receive a follow-up information from the EAS. We hope to see you again at the future EAStalk webinars and EAS events, and thank you very much and have a great day and goodbye.
Bye-bye.
Bye. Thank you.